Thank you, everyone. It's not often you get to say that you're the token male at an event, and I'm very excited to be able to say that about myself this morning. And what a beautiful setting it is. I really feel like Rome as a city, the Medici Villa, it's one of those places that teaches you what is good and possible about human endeavor. It really gives me life, so I hope you have the same spirit that we bring to the discussion today, to contemplate about how we can create a better shared future. And research, our specific topic in this session, is the core of how we improve our societies. It's how we understand ourselves, it's how we innovate, it's how we solve the biggest challenges that we all share. And funding fundamental research is one of the core responsibilities of the European Union as well. I'm no scientist. I actually stopped doing all math and science subjects when I had just 15 years. But what I am is a communicator, and one of the jobs that I had in the past was to explain and to promote the research work that the EU funds through its Horizon 2020 program. And in the course of that work, I saw a research and scientific world that, I'll admit, was very male-dominated. But it wasn't the men whose work resonated best when I was trying to tell those stories. The men were clearly very ambitious. They were good at turf wars, at pushing themselves in front of the camera. But actually, it was often the work of the female researchers that resonated best with the audiences, with the ordinary Europeans that we tried to reach out to with those stories. And in the course of that work, we also made conscious efforts to really encourage girls into laboratories, to get them into software coding, and fields that they might not ordinarily have been pushed into. And it was clear that when they were given the opportunity to do that, if they were given the space, if they were given the equal encouragement, they were just as interested and just as competent as the boys at science, technology, and so on. And all of that brings me to the three distinguished women that I'm going to invite up onto the stage now to join me here. So we have Camilla Borghese and Keifenhüller. Welcome. Patrizia Patolini Brescio. And Hanan Saab. Now, we've almost had enough of me talking, but I want to briefly introduce what connects these three women. And from a distance, what I saw connecting them was that they obviously all have a background in medical research, if you've looked at the program at all, and they're going to briefly introduce the work that they do and the impact that it has before we get into some discussion. But all three of them also clearly put the welfare of others at the top of their priority list. It's not just about personal advancement for these three women. And all three recognize the role that entrepreneurialism plays in their personal success and the shared success of our communities. And we actually, quite neatly, it's a very well put together panel, I have to say. Thank you, Chiara and Miriam. All three stages of the life cycle of an idea are represented up on the stage here. So Patrizia is involved in the really fundamental creating, the original ideas. Camilla is involved in bringing them to life, to creating and producing. And Hanan is really focused more on the delivery aspects, so getting it into the hands of individual people. Who really, really need these technologies and these ideas. So I think it's going to be a very, very interesting session. On that note, Patrizia, would you like to introduce, including via your videos, the work that you're doing in France?
Thank you very much. I would like to thank first Chiara Corazza and Clara Gremard for this invitation. Very, very happy to be here and honored to present our work in the field of early diagnosis of cancer. I was brought to devote my life to fight cancer because when I was a young doctor, I had among my first patients a patient who died in few days because of pancreatic cancer, and I couldn't do anything, no, not even relieve his suffering, anything. So this shocked me too much that I faced a choice. Either I would quit my clinical work or I would use my whole life to fight cancer. And this story I wrote in a book because from there I started to try to understand how cancer is develops and is formed. And I found that this, I worked 10 years on this. We published many papers. But then I realized that, you know, this route is too slow for a whole life to have an impact on patients with cancer. So I decided to try to understand how cancer kills. And I have a short movie I would like to show. In fact, solid cancers are cancer which develop from organs. They are the most frequent cancers, and they kill all in the same way. So the first tumor mass that you can see there started spreading cells into the blood, and those cells, they circulate in blood for years. And after years, they become capable of giving rise to what we call a metastasis, which is a second tumor in another organ, and then the metastasis starts spreading cells, and so the phenomenon is accelerating. I would like to stress two issues. One issue is that for those cells to create a metastasis is as difficult as for us to found another colony in another planet. It's really like this, because they have to become able to grow in another organ. So it's a very inefficient process, and never oncology exploited this inefficiency. So this is really what we targeted. The second point I would like to stress is that those cells, they take years, and there are challenges, very important challenges, which made that scientists could not isolate those cells and could not use them to fight cancer. Now, what you have to understand is that about 40% of all type of cancer we cannot cure nor even control. Those cancers are cancer that we discover at the metastatic stage when the metastasis is already formed. Those cancers, they start spreading tumor cells in blood, were so tiny that imaging cannot detect those cancers. So we took this angle and we started working. Circulating tumor cells have been described the first time in 1869. After that, many American scientists, they could not isolate those cells, and they could not identify them in a reliable manner. So this has been our fight. Many studies have been done on animal studies, on animal models, and I have another movie, not immediately, please. I would like to stress that we had 3 challenges. First, those tumor cells in blood are very, very rare, meaning that in 10 ml of blood, which is a very tiny volume, you have, on average, 100 million leukocytes and 50 billion erythrocytes, but just one or few tumor cells, if there are, in the blood. And so it's like finding a human in about 8 planets. That's the similarity. Second one, they are extremely heterogeneous. So they express different proteins, and it's impossible to catch them using proteins. And third one, they are not the only rare cells in blood. And so we wanted to be able to recognize them in a reliable manner without making mistakes. So 3 challenges. So what we did, that's a second movie, we invented a way to filter the blood and get out from the blood those rare cells without using proteins, so without using antibodies, just using the size of those cells. So it's this instrument and the consumable, and you can see that the blood is diluted, then loaded into the consumable, and the machine get the blood going through a filter. There are 30 parameters which have been patented in 5 patents. So as you can see, the blood flows through the filter, and the filter retains the large cells, the very, very rare cells which are in blood. And so those cells are then analyzed under microscope with the validated cytopathology way, which is the observation at the microscope, and it's possible to identify those cells without mistake. And this is because our idea is that cells from organs are much larger than blood cells, so we should be able to get them out from the blood without antibodies. But at that time, when we developed the system, the blood was considered unfilterable on a vertical manner. So right now, this system has been is used in 30 clinical centers all over the world. There are 64 independent publications showing its validity, but maybe the most important publication, it's a work which lasted 6 years, and the scientists, this is a work performed completely independently from industrial funding, so completely unbiased. And the scientists, they followed the heavy smokers without cancer for 6 years, and they applied our system called ISET and imaging. And our system could detect cancer cells in blood 1 to 4 years before the imaging found the tumor mass. So the patients have been operated immediately, and they have been cured for a cancer, lung cancer, which is the major killer, because at 5 years from diagnosis, 90% of those patients are died. So from now, we have a lot of work to do, because this is a new domain of medicine, which is able to detect cancer through a simple blood test, invasive cancer, so those cancers who are the most killer, in a very, very early manner, so giving the possibility to stop the cancer and to cure patients. So now we have a lot of work. It's not finished, but we see a path, we see a way which really can drastically reduce mortality due to cancer. That's what we did. Oh, thanks. Thank you. Thank you. Oh, well. That's too kind. Thank you.
Now, ordinarily, after a presentation of an idea like that, you think, wow, what could possibly match that? But Camilla, you're able to match it. So tell us all of the fantastic breakthroughs you've been working on.
So, first of all, it is a great, great honor to be here with you in this historical occasion, to revitalize the values of Europe. As we heard yesterday, Europe is a unique concentration of impressive, rich cultural medium. And it is up to all of us to make the most out of it. The European Commission has contributed and is contributing significantly to valorize research, because it gives founding innovative projects, is able to valorize the different expertise, and enables scientists, developers, and professionals in clinical applications to mutually learn from each other. And this is a continuous formation on the job, which is really, really impressive. And only this way it will be possible to find better solutions for the medicine of the future. Today we have a lot of new technology which enables to analyze big data and speeds up the development, and the diagnostic approach is coming reality to provide health at the end at a lower cost. And our health ministry, Lorenzin, is always saying that pharmaceutical industry actually is a culture industry, an industry of expertise, because we don't need to work to elaborate raw materials. Actually, our capital is human capital. So the Italian Biochemical Institute, in the intention of the founder, always had to preserve the goal of research to produce valid and robust results that has to be available for therapy and nothing more. And so my company is today collaborating with advanced research centers to provide a new therapeutic solution. And I will just give you some example. Recently, we joined a team of 9 partnering institutions, a multinational team in Europe, which is called Brain-Specific Modular and Active Ribonucleic Acid Therapeutics. It is coordinated by the University of Medicinal Center of Utrecht, and it targets ribonucleic acid nanomedicine against Alzheimer and spinal and bulbar muscular atrophy. It is a challenge to deliver medication to the brain that can treat neurodegenerative diseases. And this is still today an unsolved problem. So the aim of the study is to transfer efficiently to the brain, crossing the blood-cerebrospinal fluid barrier, which is a new gateway to get the medicine to the brain. And it has been discovered and applied by the University of Kent, by Rosemary. And we need to reach the brain also to put targets, ligands, on the surface of these nanoparticles to reach the brain. And this will be done by adding heavy chain only nanobodies that are smaller than the conventional antibodies. So this different delivery system, 4 different delivery systems, will be tested in vitro and in vivo, and then only the best system, the best nanoparticle, will be selected to translate in the clinical studies. And to improve, now if you can please run the little video.
Time for the video.
There is a challenge to build always a reproducible nanoparticles that has to be designed with quality by design, which means to evaluate all the critical parameters of exchanging the positive and the negative charge and the flow rate.
Our cartridge promotes self-assembly of uniform particles with controllable, reproducible, and fully scalable results. One example is solid-core lipid nanoparticles to deliver genetic medicines, which are easily made in one step using the microfluidic cartridge. The nucleic acid of interest is fed through one inlet, and the lipid components are fed through the second. Encapsulation of the genetic material in a dense lipid core occurs on the nanoliter scale in a millisecond time frame. Tuning input parameters such as flow rate ratios and lipid components enables predictable size control to optimize particles for encapsulation efficiency, targeting, and application-specific benefits. The advantages of the technology stem from the microfluidic chamber, which enables rapid mixing under laminar conditions. Proprietary architectures bring the fluids together in a non-turbulent, exquisitely controlled manner. Mixing rates are faster than particle assembly time, and every particle is manufactured under the same localized conditions. As the fluids mix, on first contact, the cationic lipids complex with the negatively charged nucleic acid to form the beginnings of the particle core. The helper lipids aggregate further as they come into contact with the aqueous solution. Finally, the PEG lipids orient themselves on the surface with hydrophobic tails pointing inwards. This process creates uniform particles and is highly reproducible. For increased volumes, the physics of one mixer can be parallelized to scale output seamlessly from milliliters to full GMP manufacture at tens or hundreds of liters, while maintaining identical manufacturing parameters.
To reach this, to produce these nanoparticles, we have to analyze a lot of data and to match, to reach the best compatibility with cells and blood. And the European Commission is investing 6 million over 5 years in this project that will enable to bridge the research industry gap and timely exploitation to the patients of these results. But the Istituto Biochimico is also collaborating with Italian excellence centers like the University of Catanzaro that has asked IBI to formulate for them a locked nucleic acid. So a locked nucleic acid is a ribonucleic acid which has been enforced with a bridge so that it can be more robust. Other projects, we have also independent projects. For example, we are working on a nanobody antibody that will make it possible for patients with breast cancer which are not responder to actual therapies to be cured. And talking about resistance and non-responder patients, today we are facing a challenge in the antibiotic treatment because of resistance of bacteria. And so, as IBI Chorus business is antibiotic sterile production, we are also collaborating to produce new advanced antibiotics, new molecules. And this is a great challenge in the battle against bacteria. IBI is almost 100 years, and in the last 80 years, it has managed by women. And I hope that we are in the best position to profit of all this cultural medium and all this new technology which are today available in Europe to cultivate our ideas and to provide the next generation therapies.
Thank you, Camilla. Now, Hanan, we'll switch focus a little bit with you, but it's an incredible story that you have to tell. So to give a little bit of the background, you trained in war in Lebanon. Your pharma company now has been severely disrupted by conflict, including in Syria. And you've got just the most huge and heartbreaking human challenges that you've been dealing with to make sure that the ideas that you work with and other ideas like these women have been producing actually get into the hands of the people who need them. Tell us a bit more about that.
Well, good morning, and thank you, Chiara and Clara, for inviting me. It's an honor to be here. I'm a third-generation pharmacist who had a business in downtown Beirut that was burned to ashes during the civil war. So as I was studying pharmacy, the business that I was supposed to take over was actually destroyed. The option for me was to actually continue my education and practice at the American University Hospital in Beirut. You'd have to realize that this was one of the most challenging areas because all militiamen and all wars were going around. So I practiced there for 5 years. I personally was several times under gunpoint to provide medications for the militiamen as well as other people who were endangered. Of course, this was a learning experience for me because I was exposed to different aspects of the business. So here I am part of completing the chain. So the drug becomes available, and Camila is one of the people that makes it available on the market. And myself, I entered into the era of marketing and distribution of the drugs. So in 1992, I decided to start my own company. It was during the war, and I have to say that, yes, I took the risk. Sometimes, looking back, I think that maybe because I was so much exposed to these adversaries, I could actually take the risk and move forward. And you might appreciate that one of the main driver for my business was availing pain medication. It was not available in Beirut because it was something like narcotics that people will be threatened to have it. And I had a relative of mine who was a cancer patient, and she was suffering big time and screaming. So this was a life-changing experience, and I vouched at the time that I will avail pain medication. It was an area that was really very active. So at that time, I came to the market, I availed these pain medications and also chemotherapy, breast cancer and others. Now the challenging part is that we are talking about very expensive medication. So you have to avail these to the public. And so you will get involved in reimbursement by the government, and you work on actually availing these products to the patients. Well, having lived in Lebanon and during different stages of the war, you might appreciate that there was all the time disruptions. I can remember, for example, during the Israeli war, of course, these attacks happen all of a sudden. So sometimes you are caught when you don't have enough stock for the patients. So what do? In 2006, the airport closed. I had to actually transport the medication from Jordan to Syria, to northern Lebanon, to arrive to Beirut. And at the time, we had multiple myeloma patients. At that day, when we were running out of stock, we had to open the box and divide the medication between the patients. And luckily, not one patient missed his treatment. This idea, I have expanded my company to become regional. So unfortunately, I have to witness what I'm witnessing today in Syria, because having lived during the civil war and having experienced all these different aspects, there was the stress of availing medications also to Syria. And you would have to appreciate that working there was much more difficult because Syria was under blockage, it was boycotted, and international companies didn't want to supply the medications because it wasn't easy. We had to get it to Beirut and transport it via land to Damascus. And it was all the time very challenging. I had to argue myself with suppliers. Of course, we're talking about a supply chain, the right temperature, the right conditions for actually availing these medications. And one would appreciate that if you have a cancer patient and you have been investing so much in the therapy of the patient, you wouldn't want to interrupt. You would want to actually allow this patient to take the full advantage of the medication. And this was the struggle. So I sustained actually the supply to these cancer patients until 2015. And pursuant to that, there was a disruption because the company refused to actually avail these medications to Syria. So we ended up getting the products to Beirut and the patients coming from there. Now, marketing this product, I just would like to give you an episode, a picture of the episode. also, the Syrian doctors were not given visas to go abroad to international conferences. So we are having the product that is availed, and you need to educate the doctors, but the doctors didn't have the chance. So what we did, and this was in 2014, we got the international speaker to Beirut, and we invited the doctors from Syria. There were 13 doctors, and we are talking about cancer treatments for adults and children. And it was heartbreaking for me to listen to the stories. Of course, we're talking about reimbursements. We're talking about availing these to remote areas. For example, in Aleppo, I remember at one time we wanted to supply a medication that was bought by the government to supply it to Aleppo, and the hospital that was using these medications was brought down. It became ashes. So it's a continuous, maybe I'm giving a perspective which is of the extreme scenario of having to work under pressure and under war times to really avail the medication. But you have to appreciate that it is a continuous chain. So they start and it comes at the end to us. And we have to avail this. You have to be innovative, you have to be creative, you have to provide everything. And because one is professional and passionate, you feel with every patient. You would want to treat this patient as if it's your own. So you get out of your way. It's very unfortunate that I see the conditions now. Of course, our business in Syria was destroyed. It brings me to the memory of the civil war and having to go through the cycle again and again. But there is always hope, and we have to sustain. I just would want to tell you one single story. I have a Syrian distributor who used to work for us, and in 2011 he arrived, he was devastated. And it broke my heart when he arrived to the office, and I felt that I want to give him all the motivation in the world. So I looked at him and I told him, listen, I have lived through the civil war and I'm still living. All that I want from you is to think long term. Everything will end. Just give it your best shot, and I will be here to avail all the medication. You can count on me. 1 year ago, he actually lives between Sweden and Damascus. His family is abroad, and he travels back and forth. He tells me, I have started a manufacturing plant. There were around 45 manufacturing plants in Syria that were destroyed. So this month, just because before coming here, he calls me and he tells me next month I'm going to start the manufacturing. And I was amazed. I was really I was really thrilled. It touched me so much, but I thought that you never give up easily. We are destined to live if we believe in destiny. So we just have to get all the maximum and get the extra mile all the time to avail things for the better of humanity. We can never escape this.
Now, I don't know if this counts as a blessing or something that's unfortunate, but those stories were so interesting and we started a little bit behind time that I let that run on a lot. So we need to move on to the next session, actually. But I'm so glad that we had that much time to hear all of your stories. And because you'll each be around for the rest of the day, then I think we encourage everyone to follow up with their questions in the coffee break and over lunch. But thank you to each of you for those inspiring stories.